Hi everyone, I'm Chi and I'm a final year PhD student in the Terahertz applications group at the University of Cambridge. Welcome to my talk today about understanding the metastability of Theophylline form 3 by means of low-frequency vibrational spectroscopy. Here's a brief outline for the day. I will start with an introduction and then I will show you the experimental and computational methods we used for this project. And I will show you the results and discussions we achieved and then end with a conclusion. So all the result discussion I show in this talk have been recently published on this paper on molecular pharmaceutics. So if you're interested in that, you're more than welcome to have a look at the paper. Let's start with the Theophylline polymorphs. For Theophylline, it actually have 5 anhydrous forms, 1 monohydrate form. For some of you who may not be familiar with the term polymorph, that means you have the same molecules, such as showing on the left side right now, we have the Theophylline molecules. And when they're packing up in a different way, they will end up into a different polymorph, and for sure the monohydrate form water is involved as well. So on the right side now shows the structure of a monohydrate form of Theophylline, and the water molecules is highlighted. There have been a long time of the publications saying that when you dehydrate monohydrate form, you will end up into a metastable state, which they call it form 3. But the structure of form 3 has never been fully published yet. So that's one of the questions we can answer from our project. And also following on, if you leave the form 3 at a certain conditions, it will in the end convert to form 2, which is the more stable and most common polymorph of Theophylline. For our project, we mainly applied Terahertz time domain spectroscopy to investigate the dehydration process from monohydrate form to the form 3. And if you're not familiar with Terahertz time domain spectroscopy, Terahertz radiation is located between the far infrared and microwave region because it's at a relatively low frequency in the spectroscopy technique. It actually very sensitive to both inter and intramolecular interactions, and it can probe the chemical motions in the crystalline materials. And also we combined with the low frequency Raman spectroscopy because for the terahertz you can, as IR spectroscopy, you can probe the motions that trigger the permanent dipole changes, while for the Raman spectroscopy you can study the motions that changes the polarizability. So with the combined method we can actually probe all the kind of motions within that low frequency range. But because of the motions really complex, we have to get some help from the simulation part to fully understand the experimental result. In our case, we used the density functional theory calculations, and two main assumptions here is that all the calculations are calculated at 0K and the atoms assumed to move harmonically, and we used the Crystal 17 software package for our project. The short video on the right corner right now is just an example to show the collective motions that we can probe in this low frequency range. Okay, so let's start from the dehydration process. We actually mixed the monohydrate form with polyethylene and compact into a pellet, and we placed the pellet in an optical chamber which is purged with nitrogen overnight, and we acquire Terahertz spectra every 2 minutes for about 17 hours. In this case, we can monitor the whole dehydration process. We actually do other mass and measurements repeats a couple of times as this as well. So we do overnight nitrogen purging, and also we try to vacuum the system as well. But all the measurements give us similar results as shown here in the figure. At the beginning, you will have the blue spectrum that has two very intense features, one at about 58 wave numbers and another one at around 95 wave numbers. And those two features, the intensity decreased drastically throughout the dehydration process, and in the end it collapsed into the red spectrum that looks very featureless, which is supposed to be very similar to the Terahertz spectra of form 3. So that also reflects form 3 can be a very disordered structure. And to fully understand what those two peaks are and what kind of motions that is, we did the DFT calculations for the monohydrate form, and we calculated both Terahertz spectra and also Raman spectra. On the left side, it shows the experimental Terahertz spectra of FM and also the calculated spectrum. But because we can't acquire the monohydrate form at the low temperature for Terahertz spectrum, as whenever we cooling down the system, we will need to vacuum the system, which unavoidably would trigger the dehydration process, so we can only give you an spectrum at around room temperature as a comparison. But even if that's the case, we can see it compared really well with our simulated result. And on the right side, we show the experimental Raman spectrum of FM at 77K, and also our calculated spectrum, and you can see it's matched very well as well. So with our simulated result, we can now see what actually happening at each peaks. So the first one showing on the top, and the second 95 wave numbers showing on the right side here. And you can see these are all Terahertz active, in another word, IR active modes. They basically reflects the motions of water molecules. So that really makes sense when you think about the hydration process, that water molecules leave the system, and you start to see the very featureless Terahertz spectrum, which because we can't probe those motions if that water already left the system. And then if you see the modes in that is Raman active, you can see they're very different from what we can see from the Terahertz active ones, because it reflects more on motions of the Theophylline molecules rather than the water molecules. It might be more obvious if you put it side by side, like the top one is Raman active, that's mainly the Theophylline framework is moving, while the bottom one is one of the motion that is IR active, which is mainly reflects the motions of water. From here, we prove that with the combined methodology, we can actually characterize all the different motions in our Theophylline system. And then it comes back to our question, what's actually the structure of form 3 is. We propose two hypotheses. So the first one is we remove the water from Theophylline monohydrate form, and then just fix the cell length and re-optimize the atomic position slightly, and that corresponds to the spectra showing here of F3A. While the second hypothesis is that we remove the water molecules from the monohydrate form as well, but we fully relax the crystalline structure afterwards and re-optimize it, which corresponds to the feature showing in the a solid spectrum as form 3b here on the left side. And then we compare the simulated result with one of the last spectrum we get from our dehydration process. So two of them are at the end of the vacuum case. So we vacuum the system for about an hour, and then we show the spectra at the very end. And then the other two is as I showed in the previous slides, that we purging the system with nitrogen overnight, and we show the last spectrum here as well. And as you can see, because the vacuum one, we didn't leave it the process for that long, so the collapsing of the two peaks is not as thorough as the two for the overnight rounds. But you can still see the decrease of the intensity and also the collapsing of the features. While the two overnight one, you can actually see it ends up to a very featureless Terahertz spectrum, which matched quite well with our F3b Terahertz spectrum. So in this case, we have some confidence to say that the form 3b is the right structure for our form 3. But furthermore, we also compare our calculated Raman spectrum with the experimental result. And here you can also see that the form 3b matched better with the experimental data, since it has three main feature regions, while the form 3a only has two main features. So from the proof from both Terahertz and the Raman side, you can see that the form 3b is the right structure for form 3. And then the last bit of a result is we spotted that after the dehydration process, we have the pellet with form 3 in it, and we just leave in the lab for about 2 days. So we measured temperature in the lab was around 20 degrees Celsius, and humidity was around 44%. So it's really like at ambient conditions. But then we run another Terahertz measurement on the pellet. It actually matched really well with the Terahertz spectrum of form 2. In that case, we proved that after 2 days at ambient conditions, the form 3 actually transformed to form 2. And here's the conclusion for our project. We used the low-frequency vibrational spectroscopy and the DFT simulations to study the hydration process of the Theophylline monohydrate form, and we also identified key motions that govern the dehydration process. And also, we showed evidence from both the Raman and Terahertz point of view of the right structure of form 3, and also we proved that the phase transition from form 3 to form 2 at ambient conditions. Because our combined methodologies actually give us many evidence of the disordered structure of form 3, so we think it's a really effective complementary method to the classic XRD method that is widely applied to study the crystallographic structure of ordered crystals, while for our combined methodology, it can also study the disordered crystals as well. And thanks for our collaborators, Adriano and Adam, and CSC for supporting my PhD study, and also Archer for supporting us with computational resources, and everyone from Terahertz Applications Group. And thank you for your attention as well.